Good morning, good afternoon, good evening for those of you joining from Africa and Asia. It's a pleasure to have you here with us. We have also the honor and the pleasure to have Rubén Rellan-Álvarez, who is a professor here at NC State. Rubén focuses on studying the genetics, physiology, and metabolic adaptations of plants to particularly abiotic stresses, including suboptimal temperatures and soil nutrient content. Rubén is going to speak about his work today, particularly in corn. And thank you so much, Rubén, for accepting the invitation and the possibility to speak to us about your work. It's great. And I'm going to give you the floor right now for you to share.
Can you hear me?
I can hear you, Rubén. Perfectly.
All right. Thank you for the invitation, Carlos. You were very persistent, so it was difficult to say no. And I changed the title slightly. I think it was a little bit presumptuous for me to give any type of lesson. So hopefully this won't be too much of a rambling. All right, so we'll get started. So as you all probably know, maize was domesticated in the southwest of Mexico. And right after domestication from the wild relative Teosinte parvilumis, maize was brought to the Mexican highlands by the people living in what is today Mexico at the time. It was brought to the highlands, and there, sorry, I have some issues here with the controls. And at the same time that it was brought to the Mexican highlands, lowland maize was also moving to South America. And there in South America, it colonized the highlands of the Andes as well. This colonization of the Mexican highlands was aided by significant gene flow between maize and another subspecies of Teosinte that had already adapted to the highlands of Mexico thousands of years before maize was brought to the highlands. So as you can see, there is a significant introgression from Mexicana in Mexican highland land races that grow in sympatry with Mexicana. And I said that there was significant gene flow between maize and highland Teosinte mexicana, but this is actually a process that still continues today. This is a field of traditional maize varieties in the border between the states of Jalisco and Michoacan. And this is a large population of Teosinte. And obviously there is exchange of pollen between these two populations. And I really like this location. We're here in this small hill. And if you look into the other side, you will see commercial cornfields where commercial hybrids are grown. So here within a distance of 200 meters, you have condensed the whole history of maize domestication, expansion into land races, and modern maize improvement. Again, this is a picture of highland Teosinte growing on the side of a commercial hybrid cornfield nearby Guadalajara. And we have known for a while that there are known introgression regions of Teosinte mexicana, like this inversion in chromosome 4. This is an inversion that has been well studied. And this inversion has been identified in highland maize growing in sympatry with Teosinte mexicana. But this inversion has also been selected in modern highland maize inbred lines from CIMMYT. So again, this is the inversion here, and this is CML459, one of the CIMMYT highland lines that they use in their highland breeding program. And very recent data from Ning Yang in the lab of Yanbin Yang and Jeffrey Rossi-Barra has shown that the extent of Mexicana introgression in maize across the Americas is much higher than we have initially thought. So here you have a quantification of a Mexicana admixture across traditional maize varieties in Mexico, Central America, and South America. And actually, if you plot the admixture proportion of Mexicana against the first principal component of genetic variation, you can see that this first principal component is basically explained by the proportion of admixture with Teosinte mexicana. And in fact, Ning Yang and colleagues have discovered that actually all modern maize has an average of 18% of Mexicana introgression. So no matter if they look at traditional land races, modern inbred lines from all over the world, they identify significant introgression from Teosinte mexicana. The only sample where they couldn't find Mexicana introgression is an old sample from Peru that is probably the result of that early expansion of lowland domesticated maize from Teosinte parviglumis that went down to South America. And indeed, this Teosinte mexicana admixture underlies phenotypic variation of important traits. So this is just an example. This is admixture for an admixture GWAS for linoleic acid, and you see that this peak coincides with a known gene DGAT1 involving linoleic acid. And this is another example of this, of a gene that has been shown that has been selected in modern breeding and impacts cob weight. But as after it moved north, maize had to adapt to longer days. And one difficulty on adapting to these northern latitudes is photoperiod sensitivity. Tropical maize is sensitive to longer days. So there had to be mutations that got around this photoperiod sensitivity for folks to be able to grow maize at higher latitudes. Photoperiod adaptation has a complex genetic basis, and it has been the subject of intensive study in the last few years. This is one of the most complete studies on this using the maize nested association mapping population. And my colleague Jing Holland and their collaborators identify major QTLs that explain this photoperiod adaptation. Among them is this gene CCN8, and work from the lab of Feng Tian has shown that Mexicana introgression in the CCN8 promoter region leads to higher expression levels of CCN8, and this helps to shorten flowering times and in the process of adaptation to higher latitudes. Again, this is an example of how Mexican Teosinte mexicana has helped maize to expand. And through this expansion today, we can say that maize is the crop with the largest geographical breadth across the world, much more than the other major grains such as wheat and rice. So as it had to adapt to this to the rest of the world, maize had to adapt to dry conditions, extreme temperatures, and all sort of abiotic and biotic stresses. In addition to that, there is a second round of selection that happens in the kitchen. Just in Mexico, there are 600 dishes that can be prepared with corn. So as you can imagine, the biochemical properties of a corn kernel that are needed to prepare a small tortilla to prepare tacos are much different than what is needed to prepare this very large tortilla that is used to prepare the typical huaraches, right? So we have adaptation to the local environment and then a second round of adaptation in the kitchen. And this has led to a wonderful variety of traditional varieties. Carlos at the beginning showed some more pictures of this phenomenal diversity. These are the number of land races that are present in countries across the Americas. And in germplasm banks, there are close to 300,000 accessions. So phenomenal biodiversity. Still, at least here in the US, we are not taking full advantage of this tremendous diversity. Most of the modern maize breeding in the US is done with a very tight breeding pool, and it's the result of two large heterotic groups, mainly the non-stiff stalks and the stiff stalks. And despite this narrow range of genetic diversity, maize breeders here in the US, they have been very successful at creating highly yielding hybrids. This is the classical one of the first ones that was derived from these two heterotic groups. And you all probably have seen a graph like this showing the continuous improvement of maize yields just with that tight genetic breeding pool. However, there are many folks that think that this is dangerous and that we should find ways of introgressing or incorporating and expanding the genetic diversity of the maize breeding pool. There are some success stories of large effect alleles from exotic germplasm, mainly with regards to disease resistance. A couple of them are HTN1 and RP1. There are also examples of how there still exists variation, in this case, a rare variation in Teosinte material that could be potentially be used to improve traits that are important for modern breeding. Again, this is work from the lab of Feng Tian, and they identify a Teosinte allele that leads to more vertical maize leaves that, as you know, has been important in modern breeding. Another example, this is work from our colleague Mayor Goodman here in NC State, looks at the identification of pollen blockers that can be used for specialty corn breeding, such as organic corn or popcorns. However, most traits in maize are highly polygenic, and they are controlled by small effect alleles. With the exception of a few variants, these effects are so small that individually they are not worth introgressing through classical backcrossing or even gene editing or even tracking on a breeding program. Some time ago, G was promised to be a good way to identify some of these polygenic traits that could be introgressed, but it hasn't been that successful. And as early as in 1993, folks like Simmons, they were promoting the idea of base broadening or incorporation rather than introgression to introduce exotic germplasm material in a new environment. So the idea here is that you can take an exotic germplasm and through several rounds of selection, adapt it to a new environment, make it competitive in that new environment, and that allows you to increase the genetic pool that is available for you to breed with. The problem with this approach is that it takes a long time, years or decades, and it's not always easy to fund this type of approaches. More recently, my colleague Jim Holland has promoted the idea that we could use genomic selection on purely unadapted exotic populations to first do genomic selection for adaptation to a new environment, and then for combining ability before we can take advantage of these exotic materials. There are examples of these approaches. One of them is the successful use of Ecuador 573. This is a highland landrace from Ecuador. The CIMID successfully adapted to the highlands of Africa, and this is now the base of the highland breeding program of CIMID in Africa. Another example is the indefatigable work of Dr. Mayor Goodman here in NC State. As you know, Mayor Goodman has been a big proponent of the need of introducing tropical diversity in U.S. maize breeding programs, and many of the lines that came out of his program are purely tropical materials that he adapted to temperate conditions through multiple rounds of selection. An intermediate approach is also the work of Matt Krakowski here at USDA and the genetic enhancement of maize program, where tropical varieties are crossed with proprietary inbreds from industry, and then they are selected for high yield in U.S. conditions. In our own work, we have been interested for a while in local adaptation of highland maize. We think that highland maize is a good model for adaptation to low temperature and early season drought. In typical highland conditions in Mexico and other countries in Latin America, because of the low GDU accumulation, farmers have to plant very early in the season, as early as in February. This early planting maize has to wait for rain to arrive in May, and then is harvested very late, around October or November. Carlos mentioned that we're also very interested on adaptation to soils with low nutrient availabilities. The highlands of Mexico are very interesting in that regard. We have a chain of volcanoes that cross Mexico, the Trans-Mexican Volcanic Belt, and a lot of these highland volcanic soils have low soil phosphorus availability. I'm in the biochemistry department, so we are interested on understanding how changes in the metabolism help maize adapt to these conditions, and we are particularly interested on the role that glycerolipids play on this. So phospholipids are a class of glycerolipids, and they are essential building blocks of plant cell membranes. But there are other types of glycerolipids that are distinct from phospholipids because they contain a different polar head other than this phosphorus group. So these are mainly galactolipids and sulfolipids. And phospholipids, because they are essential building blocks of plant cell membranes, they take up around a quarter of the total phosphorus in a typical plant cell. And what happens is that in cold conditions, plants tend to increase the concentration of phospholipids and reduce the concentration of sulfolipids and galactolipids. And this is because plants they want to maintain membrane fluidity in low temperature conditions. And phospholipids in general, they are more polar than these other types of glycerolipids. However, under low phosphorus conditions, plants tend to degrade phospholipids, particularly in older leaves, to make those phosphorus groups available for other more important functions of the cell, such as the production of RNA for ribosomic RNA for protein production. And these phospholipids are replaced by other glycerolipids, such as sulfolipids and galactolipids. In addition, phospholipids have been shown to regulate flowering time through binding and releasing of florigen at different stages. And it has been shown that in particular, certain species of phosphatidylcholine can bind to flowering locus T, which in maize is CCN8. On the meristem of the cell, the hypothesis is that this binding helps to recruit that CCN8 transcription factor into the nucleus. And there, once it's recruited into the nucleus, it can accelerate flowering time. So we have been working with this hypothesis that says that phospholipid metabolism was under selection and is important for maize adaptation to highlands. So from now on, I will tell you the results of a paper that we have recently published. So the first question that we ask is, well, is phospholipid metabolism under selection in highland maize? And to do this, we use this population branch excess statistic that is based on the use of FST, or a measure of genetic differentiation. And it basically, so what we did here is we use whole genome sequencing of 4 highland maize populations, and we compare it with a couple of lowland populations from Mexico and South America. So when we did this analysis, we discovered that pathways involved in phospholipid remodeling, having repeated targets of selection across all 4 highland maize populations. So these pathways are involved in breaking one of these fatty acids from a phospholipid molecule, breaking the phosphate group, etc., etc. This is, I'm focusing here on a couple of such genes, LPCAT and HPC1. LPCAT takes one fatty acid and one lysophospholipid and makes a phosphatidyl choline. And this HPC1 gene is a phospholipase A1, and it does the opposite reaction. It will take phosphatidyl choline and it will chop off one of these fatty acids and it will produce a lysophospholipid. So this was very interesting to us. We further explore selection to highlands using this approach called PC-Adapt. So on PC-Adapt, you take genetic information, and here genetic markers, and here we use around 2,000 land races from Mexico. And PC-Adapt identify adaptive loci by evaluating how much they contribute to major axis of genetic variation. So this is a first principal component analysis. What you can see here is that the first principal component is polarized by elevation of origin of these land races. So then you can use correlations of genetic markers with the first principal component to identify loci that are under selection with altitude, because again, this first principal component is mainly polarized by elevation of origin of the land races. And then we focus on HPC1 and LPCAT1, and we could observe that SNPs that fall on the CDS of these genes are outliers on this first principal component of PC-Adapt. If you take a look at the minor allele frequency changes in relationship with elevation, you can see a very strong pattern as well. So this is all nice, but then we wanted to evaluate what is actually happening with these compounds. And to do that, we perform 2 common garden experiments in the highlands and lowlands of Mexico, and we evaluated around 120 land races from Mesoamerica and South America. Half of them are highlands, half of them are lowlands. So I'm not going to explain all the results, but something that struck us is this what I'm highlighting here, and this is showing us that highland Mexican land races have a higher LPC ratio than lowland land races, and this is particularly true when we do these experiments in highland environment. At the same time that we did this experiment, we also evaluated B73 by Palomero Toluqueño biparental mapping population. B73 is a temperate inbred line. Palomero Toluqueño is a highland popcorn adapted to the Central Valley of Mexico. So you can see how Palomero Toluqueño is adapted to its local environment, while B73 struggles to make it in this environment. It actually takes 150 days for B73 to flower in this environment that has very low growth degree unit accumulation. So we grew these biparental populations side by side with that diversity panel. Again, this is a picture of Palomero toluqueño expressing typical traits of highland maize varieties. They're highly pigmented. They have a lot of hairs on their stem. So we took samples from these biparental population lines, and we measure phosphatidylcholines, lysophosphatidylcholines, and the ratio between them. And we identify a major locus at the beginning of chromosome 3. And at the peak of this QTL is where HPC1 is located. So if we look at the effect sizes of phosphatidylcholines, lysophosphatidylcholines, and the ratio between them, we can see that at that QTL peak, that major QTL peak, if the recombinant inbred line is homozygous for Palomero toluqueño, it has high levels of PCs, low levels of LPCs, and therefore a high LPC ratio. And we observe the opposite pattern if those recombinant inbred lines are homozygous for B73. Now, if you remember, HPC1 is annotated as a phospholipase A1 that converts phosphatidylcholines into lysophosphatidylcholines. So if we look at this pattern, we hypothesize that the Palomero toluqueño allele is a loss of function or misfunction of HPC1 because we have higher levels of PCs than LPCs than when we compare it with B73. And in fact, we generated a CRISPR-Cas9 mutant of HPC1, and we observed that the null mutant of HPC1 phenocopies the metabolic phenotypes of Palomero toluqueño, further supporting the hypothesis that the allele of Palomero toluqueño is a loss of function of HPC1. So we then went ahead and sequenced the Palomero toluqueño allele, and we didn't find anything major such as a big deletion or anything like that, but we identified several non-synonymous SNPs that could affect the enzyme's function. In particular, this single SNP here that leads to a conversion of isoleucine to valine in Palomero toluqueño. This is not such a big change, but this allele is located in the flap lead of the phospholipase, and the flap lead is what allows the phospholipase to accept or reject the substrate, which in this case would be those phosphatidylcholine molecules. So because of the location of this mutation, we think that this may be one of the causes explaining our phenotype in Palomero toluqueño. So then we went back to that collection of whole genome sequences from Mexican highlands, Guatemalan highlands, Mexican lowlands, Andes, and South American lowlands. And we explore whether those land races contain the B73 allele that I or the Palomero toluqueño allele, that V. And what we can observe is that Palomero toluqueño allele seems to be fixed in Guatemalan highlands, Mexican highlands. It's also present, although it's not fixed, in the Southwest U.S. material. The Southwest U.S. material is known to be the result of highland route from Mexico into that area. So it makes sense that it also carries that highland Mexican allele. In Teosinte mexicana, we also observe the Palomero toluqueño allele, and in Parviglumis, it's mostly Parviglumis carries mostly the B73 allele, but there are some Parviglumis accessions that still carry the Palomero toluqueño allele. So then the question there was, well, is this Palomero toluqueño allele the result of selection on a standing variation from Parviglumis, or the result of Mexicana introgression? So to answer that question, we did an ABA-BABA test, and without going into details, we could see that this Palomero toluqueño allele is in fact the result of introgression from Teosinte mexicana. And then we went and explored the presence of this Palomero toluqueño haplotype in modern inbred lines, and we could see that the Palomero toluqueño haplotype, including here Palomero toluqueño, and this is one of the Teosinte mexicanas. So this haplotype is conserved in northern US and Canadian and also European flints. We took advantage of a large RNA-seq experiment where an RNA-seq was performed in the 282 panel. The 282 panel is a collection of inbred lines developed here in NC State some years ago, and it has been widely frequently evaluated. So then we can do this sort of analysis. We can see if there is correlation in the levels of expression of HPC1 and flowering time. And what we can see here is that those inbred lines that carry the Palomero toluqueño allele have low expression of HPC1, and they also have shorter flowering times. In fact, when we went and explored possible hits of flowering time using the published Nextera Association mapping population, there is a hit in the coding sequence of HPC1 for days to silking, and this hit explains around 8 hours of flowering time variation on the Nextera Association mapping population, which for maize is pretty decent. And we also found that the flap lead domain marks a big recombination point between two strong linkage disequilibrium blocks. So we're now exploring whether in addition of this allele that may affect the protein activity itself, there are other alleles on the promoting region that may be affecting the expression levels of HPC1 as well. Then we asked the question, well, if this was selected in Mexican highlands, does it actually confer a fitness advantage to Mexican highland maize in highland conditions? So to try to answer that question, we used data from a large a common garden experiment that was performed by CIMMYT across 23 field trials, and they use 2,700 land races that were test cross to produce F1s, and those were evaluated. So all the genotype data and the phenotypic data is available for this large experiment. So when we look at that data, we found that HPC1 is a strong genotype by environment outlier for flowering time and other fitness traits such as cob weight. And I'm marking here the effects of inversion foreign that I talked about earlier. So then you can explore what is the effect of the Highland allele of HPC1 depending on the elevation of these yield trials. And you can see that for things like days to anthesis and fresh ear weight and other fitness traits, we can observe a strong G by E effect in such a way such as that when you carry the Highland allele of HPC1 and you are grown in lowland environments, it leads to a delay of flowering time. But we see the opposite effect when you carry the Highland allele of HPC1 and you're grown in highland environments. So with that, I'm going to finish. This last part was published on this paper, and the result of wonderful collaborations with many colleagues, but the folks that really led this work were Alison Barnes, Carla Juarez, and Fausto Rodriguez Zapata in our lab. I want to thank all our collaborators across different labs and the folks in my lab. And with that, I'll be happy to take any questions.
Thank you, Rubén. Thank you so much for your wonderful seminar. Please post your questions in the chat as we can relate those to Rubén. I'm gonna start with a question, Rubén, from your early part of the presentation when you show the integrations from Mexicana. Were those integrations, you think, sort of a founding effect, or they were repeated? What is the spread of Mexicana? It's just very localized in Mexico, or is it throughout?
Yeah, correct. Mexicana is mainly localized to the highlands of Mexico. There are 4 distinct Mexicana land races, right? Chalco, Mesa Central, Nobogame, and Durango. So Nobogame and Durango, they are farther north. Mm-hmm. But this is the area where Mexicana currently grows, naturally grows, obviously. Mm-hmm. Mm-hmm. So you can read the paper from Jeff Ross Ibarra, but their hypothesis is that after domestication from Parviglumis and expansion to the highlands, there was again significant introgression from Mexicana, and that introgression and admixture with Mexicana is what truly made maize so successful, mm-hmm. right? And again, they have found very high levels of Mexicana introgression, much more than we originally thought, and no matter what material they look at.
Okay. I have a question here from Roshan. It's a very satisfying story. Were you able to round up any trans EQTLs that are worth looking into in terms of HPC?
Trans EQTLs. No, we haven't started to look into that, we have some indication that there may be alleles on the promoter of HPC1 that may be affecting the expression of HPC1. It seems like, at least in cold conditions, this is preliminary data that is not on the paper, but in cold conditions, Palomero toluqueño doesn't seem to be able to express HPC1 at high levels, while temperate materials such as B73 and other temperate inbred lines, we see high upregulation of HPC1. Okay, so those patterns of expression would fit also with the phenotypic results that we see, okay, which is high levels of PC in highland materials and low levels of lysophosphatidyl-choline in highland materials. Again, that can be achieved through reduced expression of HPC1 or through misfunction of the protein itself.
Thank you, Rubén. Xu Chen asked if it says it has been reported teosinte has greater nitrogen use efficiency than modern maize. And he's wondering if you have observed any differences in nitrogen-related pathways besides the phospholipids for Mexican adaptation to highlands.
Thank you, Chuye. Nice to see you here. No, we have not done that yet, but we're planning on doing it. I didn't have time to speak about that today, but we're embarking on a large project looking at nitrogen use efficiency and other important traits related with nitrogen. And as part of that project, we are in the process of developing new mapping populations that include a variety of teosyntes backcross into B73, and those includes all these four races of teosinte mexicana that I talk about. So we're very excited about what may come out of that work. Yeah.
Good. So stay tuned for another seminar, Rubén. Isaac is saying, you mentioned that incorporation is better than introgression when it comes to adoption of exotic species to a new environment, adaptation probably. Can you please provide more details to that information?
Thanks for your question, Isaac. I'm not claiming that is necessarily better, right? Simmons, Mayor Goodman, Jean Holland. I think what the idea is that if you want to bring in traits that are largely polygenic and that may be difficult to incorporate through classical backcrossing introgression. This idea of taking exotic populations and taking the time through several rounds of mass selection or using modern tools such as genomic selection, taking the time to adapt those exotic populations to a new environment, it can be a very useful way to then more easily be able to figure out the actual genetic value of those exotic materials. Okay, and I don't know if Jim is in the talk, but he can correct me if I'm wrong.
I see Jim being present here. Any other questions? I think we have one more from Meng Yang. Nice presentation. Is the tolerance to coast trait associated to the susceptibility to phosphorus deficiency? I cannot read this well here.
Yeah.
Can you read it? Yeah. Okay.
Yeah. Thank you, Menyan. What we have observed, but we actually need to do those experiments that you are hinting at there in your question. What we have observed is that the mutants of HPC1, at least when grown in control conditions, they don't show major differences with the wild type with respect to phosphorus levels. Okay, but what we do know is that materials adapted to these highland environments of Mexico and these acidic soils that have low phosphorus availability, we know that these materials have lower levels of phosphorus in the kernels, higher phosphorus use efficiency, etc., etc. The possible role of this particular gene that we have identified in that phosphorus and cold trade-off, we still have to do those experiments.
Any other questions? Yeah.
This is a message for the folks in the lab that hear me saying this several times.
Take note. I had a related question to the one I asked you before. Is it people studying the effects of modern corn on Mexicana current evolution? Because it looks like from the photos you showed, that's a very active exchange, and we are probably
there is naturally, I think that there is much more work done on the possible effect of Mexican introgression into maize than vice versa.
Right.
Related to this, I mentioned earlier that the natural environment of Mexicana is restricted to the highlands of Mexico. But in the last decade or so, in farmer fields in France and in Spain, they identify some weedy corn that was starting to become a real issue for them. And recent analysis have shown that weedy corn is actually Mexicana, okay? And it has probably been able to adapt to those conditions and mix well with maize through changes actually in CCN8.
Well, it happens in rice too, isn't it? With wild rice.
Yeah, weedy rice is much more studied.
Yes, exactly.
Mexicana likes to grow on the sides of the fields. It's very difficult to identify until it actually flowers, right? Yeah. But much harder when they are small seedlings. Parvigloomies tends to grow in more natural environments, not so much cultivated environments. But yes, less research has been done on the effect of maize into Mexicana. There are some work in Mexico done on possible escapes of transgenes into teosinte in general, right?
Good. Okay. Roshan has another question here. Says, are there any observable nutritional differences in corn for highland versus lowland as it relates to this adaptation? You mentioned phosphorus content is one.
Yeah. Again, phosphorus in Mexico, these areas of low phosphorus availability coincide with high elevation areas, and Fausto has done some analysis that shows good correlation restricted to Mexico, but good correlation of soil phosphorus availability and elevation. Okay, obviously it depends a lot on the location, right? But there is a connection there with elevation and phosphorus availability. Perennial theosynths. There are a couple of perennial theosynths, perennis, which is tetraploid, and diploperennis, which is diploid. There's a lot of interest from folks interested on nutrient recycling, particularly nitrogen, to see if there are typical perennial traits that could be exploited to improve on nitrogen and other nutrient recycling at the end of the season in corn. Not nutrient related, but there are other subspecies of Teosinte, such as Nicaraguensis luxurians, that have shown some interesting traits with regards to tolerance to submergence. Okay, because they naturally grow in areas where they have to deal with a lot of rain and large periods of time where they are submerged.
All right, Rubén, I'm going to take the floor from you. And thank you so much for a wonderful seminar, Rubén, and thank you for the attendance to the seminar for everybody. Just wanted to also thank Brandon and Tiago for their help in organizing the seminar. We have the last seminar of the series next Thursday, and because it's going to be presented by Dr. Robert Henry from the University of Queensland, who is about 14 hours ahead of us, we have decided for this particular location to host it 2 hours earlier. We're going to announce this again tomorrow and next week to remind people that next seminar on May 4th is going to be at 9 a.m. And Dr. Henry is going to be talking about domestication of species using molecular means. I think it's going to be an amazing seminar, also as all the seminars have been this series. Thank you so much, Rubén. Thank you for everybody who attended. I hope you guys have a wonderful weekend, and look forward to seeing you next week. Thank you.